Nephronophthisis, which means nephron wasting, and medullary cystic kidney disease, which refers to fluid filled sacs in the medulla, are two kidney diseases that share some similar features. They're both genetic, they both affect the nephrons, and they both can lead to kidney cysts and renal failure over time. To help understand these diseases, let's first take a zoomed in look at a nephron and talk about how it works. Alright, so the outer layer of the kidney is called the cortex, and this is where the glomeruli live, which is where blood is initially filtered into the nephron, as well as the proximal convoluted tubule, where some of the filtered substances are reabsorbed back into the body. The filtered substances, or filtrate, that don't get reabsorbed then moves down the medulla via the descending and ascending parts of the loop of Henle. The filtrate then goes back to the cortex just briefly in the distal convoluted tubule, and then returns back to the medulla in the collecting duct. Zooming back out a bit, the collecting ducts in each region of the kidney, called a renal pyramid, converge on the renal papilla, which dumps fully formed urine into a minor calyx. From there, the urine goes into the major calyx, and soon after it goes into the ureter and the bladder. And finally, zooming back in, surrounding each nephron's tubule is the tubular interstitium, a hypertonic environment optimized to help reabsorb water and other substances from the tubules. Alright, so in nephronophthisis, which presents in childhood, the tubules atrophy and the interstitium gets infiltrated by macrophages and becomes fibrotic. Inflammation of the tubules and the interstitium qualifies nephronophthisis as a tubulointerstitial nephritis. But don't confuse this with nephritic syndrome, which is where red blood cells and protein escape in the urine as a result of damage to the glomerulus. In nephronophthisis, the affected tubules lose their ability to concentrate the urine by reabsorbing water and other substances back to the body. So urine ends up being more dilute than usual, which leads to polyuria, excessive urination, and therefore polydipsia or excessive drinking. Sodium wasting also happens, which is where excess sodium gets excreted in the urine. But proteinuria, or proteins in the urine, as well as hematuria, or blood in the urine, typically aren't seen in nephronophthisis. Later on in the disease, the glomeruli can become sclerosed, or scarred, and cysts might pop up in the medulla, particularly in the corticomedullary junction, which is where the cortex meets the medulla. Over time, this leads to renal insufficiency, or poor renal function, which can cause uremia, or too much urea in the blood, and anemia from a failure of the kidneys to make erythropoietin. Eventually, this leads to renal failure, usually in the teenage years. Nephronophthisis is broken into subtypes by the age of onset, like for example infantile versus juvenile, or by the gene that's mutated, of which there are over a dozen possible mutated genes, all of which are inherited in an autosomal recessive fashion, though the most common is NPHP1, which codes for nephrocysteine 1 protein. Most nephronophthisis genes encode for and therefore affect proteins in primary cilia or a related organelle, the centriole. A primary cilium is present on almost every human cell, and it's a tiny appendage that projects outside the cell and conveys sensory input to the inside of the cell. The centriole, on the other hand, is the main component of the centrosome, and helps organize the mitotic spindle and microtubules during cell division. When there are defects in these structures, the epithelial cells lining the kidney are seriously affected, and this leads to the tubule atrophy seen in nephronophthisis. Primary cilia and other parts of the body rely on these proteins too, which explains why other organ systems can be affected by nephronophthisis. For example, it can cause liver fibrosis or situs inversus, where the major visceral organs are reversed from their normal positions. On the flip side, medullary cystic kidney disease, or MCKD, has similar kidney changes as nephronophthisis, like tubulointerstitial fibrosis and inflammation, tubular atrophy, cysts in the medulla and corticomedullary junction, and eventually glomerular sclerosis. MCKD is also clinically similar, with progression to polyuria and polydipsia from poor urine concentration, as well as salt wasting and eventually renal insufficiency and renal failure. Some differences are that MCKD starts in adulthood and doesn't have symptoms in other organs except for hyperuricemia because of a failure to excrete uric acid, which causes gout. Also, there are two types of medullary cystic kidney disease, one called MCKD1, which is caused by mutations in the MUC1 gene, and the other called MCKD2, which is caused by mutations in the UMOD gene. And both of these conditions are autosomal dominant. For diagnosis, nephronophthisis and medullary cystic kidney disease might be suspected when an individual has polyuria or polydipsia as an early symptom, or if there's a family history of the disease, or if there's a finding on imaging like kidney cysts. To confirm the diagnosis, a renal biopsy or genetic testing can be done. And based on the severity of the disease, treatment primarily involves dialysis and kidney transplant. Having said all that, it's important to remember that renal cysts can be caused by a lot of other diseases, and they can also just be simple renal cysts, which are common age-related findings that tend to be in the cortex and don't usually cause any problems. Okay, as a quick recap, nephronophthisis and medullary cystic kidney disease are genetic disorders that affect the renal tubules and interstitium and can lead to cysts and renal failure. Nephronophthisis presents in childhood, is autosomal recessive, and can involve other organs, whereas medullary cystic kidney disease presents later on, is autosomal dominant, and affects only the kidney. Thanks for watching. You can help support us by donating on Patreon or subscribing to our channel or telling your friends about us on social media.